I am Edward Dennis, and I welcome you to Lecture 3 on cholesterol regulation and homeostasis. This lecture will be divided into two parts. In Part A, our focus will be on cholesterol itself, the molecule, with its special relevance to cardiovascular disease, its biosynthesis and regulation, and the effects of oxidation and esterification to make new and different molecules. In Part B, we will focus on its inhibition. First, direct inhibition of its synthesis by pharmaceutical agents, and then second, its more complex regulation by SREBP and LXR and RXR as well. The learning objectives for this lecture on cholesterol regulation and homeostasis are listed here, and I ask that you read these over prior to the lecture. Reading for this lecture is stated here. First, we should ask, why do we care about cholesterol? Well, you can see from this graph that the death rate due to cardiovascular disease increases dramatically as serum cholesterol levels increase. And if we use a kind of round number of 200 milligrams per deciliter, one sees that above that, there's a very proportional increase in disease. Heart disease is the #1 killer in the U.S. It is today largely preventable. It's strongly linked to cholesterol. And now, with new drugs, it is possible to dramatically decrease cholesterol levels in the serum. Let's pause for a minute and look at the big picture of lipid biochemistry. Fatty acids are broken down to acetyl-CoA and then enter the TCA cycle to generate ATP, sources of energy. Some portion, under certain physiological conditions, are converted to ketone bodies. Acetyl-CoA is converted into fatty acids for the storage of fat, and these fatty acids are esterified into membrane phospholipids and triacylglycerols. The acetyl-CoA, which is at the center of metabolism, is also converted under certain physiological conditions to cholesterol, and that is our focus today. Cholesterol being such a central molecule to metabolism and disease, it's good to sit for a moment and just look at its structure. It consists of 4 rings, which make it somewhat planar. It's completely hydrophobic, with the exception of the beta-3 hydroxyl group on the lower left-hand corner. And sometimes we refer to the C8 chain as the chain in the upper right-hand corner off of the 5-membered ring. That single hydroxyl group can be either in the alpha or beta position as illustrated. Natural cholesterol, and what we refer to as cholesterol, always is in the beta or up position of the 3 carbon on that first ring. And when one draws a more stereochemically correct diagram or picture of cholesterol, you see how flat and planar that ring structure is with the single hydroxyl in that beta position on the backbone. Synthesis of cholesterol from acetyl-CoAs is a 25-step pathway. The essential elements are shown here. 3 acetyl-CoAs combine together to make hydroxymethylgluteryl-CoA, known as HMG-CoA, and a key enzyme in its control and regulation is the HMG-CoA reductase, which converts it to mevalonate. Mevalonate is converted to isopentyl pyrophosphate and then dimerizes and further polymerizes to make squalene, which when it cyclizes becomes cholesterol. The important first steps are shown here. 2 acetyl-CoAs are condensed by the enzyme thiolase, which makes acetoacetyl-CoA. HMG-CoA synthase, another important enzyme and sometimes regulated, adds a third acetyl-CoA unit to make 3-hydroxy-3-methylgluteryl-CoA, or HMG-CoA. This key enzyme, the so-called regulated step, is HMG-CoA reductase acting on HMG-CoA to produce mevalonate, shown in the lower right. Mevalonate itself undergoes several more reactions, resulting in a key intermediate 3-isopentenyl pyrophosphate, shown in the upper right. There are 5 carbons in this key intermediate with the pyrophosphate on it is highly energetic and reactive. As I indicated, the key component of the isopentyl pyrophosphate is that 5-carbon backbone, which is denoted as an isoprene. Isopentenyl pyrophosphate is dimerized, and then those dimers come together to polymerize to form squalene, which is made up of 6 isopentyl pyrophosphate backbones, as shown. The dashed line indicates where the subunit isoprenes are located. The isoprene has this unique 5-carbon arrangement with a double bond, and although further reactions can occur to it in some molecules, one sees isoprene backbones in many different important lipids. In particular, the fat-soluble vitamins, vitamin A, vitamin D, vitamin E, and vitamin K, all have isoprene backbones and are derived by similar reactions to that of cholesterol. While cholesterol is very important in metabolism, interestingly, much of the cholesterol exists as a cholesterol ester and not as free cholesterol. This is accomplished by the enzyme acyl-CoA cholesterol acyl transferase, shorthand is ACAT, which is a intracellular enzyme that takes a free fatty acid as a CoA derivative and acylates that 3-beta hydroxy position on cholesterol. And we have already encountered ACAT in our discussion of lipoprotein receptors and the fact that ACAT intracellularly converts free cholesterol to cholesterol esters. Another key enzyme in making cholesterol esters is LCAT, shorthand for lecithin cholesterol acyltransferase. This enzyme takes free cholesterol and adds to it a fatty acid from the middle or SN2 position of lecithin, a common name for phosphatidylcholine, to make as products lysolecithin or lysophosphatidylcholine and cholesterol esters. LCAT generally resides on lipoproteins and accomplishes the conversion of free cholesterol to cholesterol ester. Interestingly, 2/3 of plasma cholesterol is generally found as cholesterol esters, and this is accomplished by the enzyme LCAT. This is a scheme that I introduced in an earlier lecture on lipoproteins, but now we can look at it from the broader perspective of cholesterol regulation. So HMG-CoA reductase is that key rate-controlling enzyme in the synthesis of cholesterol. It is regulated at the hormonal level by glucagon, which downregulates or turns off the HMG-CoA reductase, decreasing the amount of cholesterol synthesis. In addition, cholesterol ester taken up from lipoproteins also downregulates the HMG-CoA reductase. In contrast, insulin is a promoter of cholesterol synthesis and activates HMG-CoA reductase products. Intracellular synthesized cholesterol downregulates the LDL receptors, and by decreasing the number of receptors, the cell will take up less extracellular cholesterol. And as we previously described, cholesterol is converted to cholesterol esters by the enzyme ACAT, which is intracellular. So far, I have stressed the important role of cholesterol and cholesterol esters. But cholesterol is also the precursor of many other important biological molecules, most particularly the steroid hormones, and cholesterol being converted to pregnenolone and progesterone and cortisol, corticosterone, testosterone, and many further derivatives of these as well, each of which plays critical roles in many processes, particularly reproduction. Another central role of cholesterol is as a precursor of the bile acids and bile salts. The key first step in bile acid production is the activation of the 7-alpha hydroxylase, which adds a hydroxyl group at the 7 position, and with additional hydroxylations and oxidation of that C8 hydrocarbon chain produces cholic acid, a key and central bile acid. Often the bile acids are found conjugated to amino acids, and shown here, glycocholic acid is a bile acid, bile salt, that has glycine conjugated to it. Now these bile acids are made in the liver. They are oxidized forms of cholesterol, and so they are a way of disposing of cholesterol as well. They're secreted into the bile ducts, from where they further are released to the GI tract, and in that process they serve as biological detergents or surfactants to help to solubilize the lipids that are taken up from dietary sources. Some of the bile acids are reabsorbed through an extrahepatic circulatory system, and some are secreted. Thus, an important take-home message from this is that when we're through with molecules that we've needed, we often get rid of them by oxidation. And cholesterol is an excellent example of that. This is a resultant oxidation of cholesterol. The net result is that cholesterol is decreased in the liver, and that decrease in cholesterol in the liver causes the upregulation of LDL receptors, so it creates more receptors on the surface, a removal of circulating LDL, and an overall lowering of plasma cholesterol. Thus, in summary, the circulation of cholesterol and its formation of bile acids is a very key regulatory element in the control and regulation of overall cholesterol levels.